Okay, should we continue our discussion? Okay, so one last question here. Okay, everybody got a chance to answer? Okay, so hyperhomocystinemia, I think, is the best answer here. Would the patient have a macrocytic anemia? I don't think so. Yeah, I think the folate, the thymine cycle should be fine. Okay. If you have any questions on folate or B12 or S-adenosyl methionine, go ahead and shout them out. Otherwise, I'm going to queue up the next slide. Okay. We're back. All right, so for the next couple hours, or perhaps less, we'll talk about nucleotide metabolism. And we'll talk about the basic pathways of synthesis of the nucleotides and their degradation. And we'll talk about the disorders therein. Okay, so first we'll just have a little bit of an introduction. Then we'll talk about the purine synthesis, salvage pathways, and degradation pathways. Then we'll talk about the pyrimidine synthesis and degradation pathways. And then we'll talk about a few clinical cases. Okay. Oh, oops. Hang on just a second. I got to reset my slides here. All right, should have done this over the break. So. All right, anyone from out of town?
That's why they're not going.
Yep. Okay, should we review some stuff from last week? Got a few questions from urea cycle stuff and then a few questions from folate. And for this question, I guess it's a little not clear, but looking at the direct donors of the nitrogen to the urea molecule. The substrates. Okay, good. So carbamoyl phosphate synthase 1 uses ammonium, and then argininosuccinate synthase uses aspartates. Okay. This should be argininosuccinate, so missing a couple of letters there. If that helps, I'm not sure. Oh, very good. So this reaction, we're going to see this react, or a very similar reaction to this in nucleotide synthesis. So revisit that one. Okay, super. So ornithine transcarbamylase, that enzyme is right below the formation of carbamoyl phosphate. Okay, so further on to this topic. Okay. Very good. So carbamoyl phosphate synthase 1, that's the enzyme that makes carbamoyl phosphate. So if you're not making it there, it can't leak out into the pyrimidine pathway. Okay, serine, just by the molar amount of carbon that's going into tetrahydrofolate, serine is the major donor. And when serine donates a carbon to tetrahydrofolate, what does it make?
It makes one of them. I think it could be A through D. Oh, never mind. Yeah, it was not as hard.
Okay, so you know there's got to be glycine in there, right? So look for the one with glycine. So N5, N10 methylene tetrahydrofolates is the best answer here, and glycine. And then the glycine cleavage enzyme can make another N5, N10 methylene tetrahydrofolate. Okay, this question is a little, okay, good. Best answer would be deoxyuridine monophosphate. The enzyme isn't inhibited by 5-fluorodeoxyuridine itself, right? That 5-fluorodeoxyuridine has to be modified by the addition of the ribose sugar and then phosphorylation, right? So 5-fluorodeoxyuridine, that's a prodrug. So this isn't the most elegantly written question. You want to talk this out with your neighbors? This is kind of maybe a difficult one.
I guess as well.
I know you did too, but I was correcting when I get corrected.
Examples from the end of the last class, all those lab values are the same as the last.
I think
Okay, we got a good discussion here. Okay, parenteral cobalamin. Everybody agree with that? So the clinical picture looks like pernicious anemia, right? He's got let's see, decreased hematocrit and hemoglobin with an increased mean corpuscular volume. So there's your macrocytic anemia. And then he's got the peripheral neuropathy, right? So neurological feature. So that's kind of a consistent with the pernicious anemia. But he's omnivorous and he takes a multivitamin daily, right? So it's not like he's a, vegan or wouldn't have vitamin B12 in his diet, right? So of the choices then, I think the parenteral cobalamin would be the best choice. And if the symptoms resolve then the implication is that the child had a problem with absorbing dietary vitamin B12. And this would be consistent with a inborn error of metabolism that results from mutations in the gene that encodes for intrinsic factor. Now recall one of the major causes of pernicious anemia is autoimmune destruction of the parietal cells. So that would be unlikely in a 4-year-old. So that's something that generally manifests in adulthood. But you would get basically the same thing if you had an inherited loss of function mutation in intrinsic factor gene. Okay, it wouldn't manifest immediately after birth because the body has a huge store of vitamin B12 in the liver. Okay. Okay. Oh. I don't know if I can fix that. Okay, so this is the same stem. Okay, no, it had to be on one end or the other, right? So this is the methyl trap hypothesis. Okay, if you don't have vitamin B12 around, then you don't have any fate for the methyl tetrahydrofolate. And your whole folate pool gets trapped there because you can't oxidize the methyl on methyl tetrahydrofolate. Okay? Nope. Excellent. Got to help each other out here. Okay, are there questions on folate or B12 or S-adenosylmethionine? Yes.
On that last case, what was the cause of the diarrhea in that inborn error?
With the cystathionine synthase deficiency?
No,
Or the,
just that last one.
okay, I'm gonna, this one?
Yeah, I think so.
I don't know the answer to that. I mean, if you had like some other, like an infection or Crohn's disease or something that would be associated with diarrhea, you could also get a malabsorption deficiency that could manifest as a B12 deficiency. I apologize, but I can't think of a direct link on that one. Good question. Okay, so perhaps we should switch gears subtly and talk about nucleotide metabolism. Okay, so nucleotide metabolism is on the Atlantic seaboard, I guess, going from Maine down to Florida and then the Gulf Coast down there. Okay, so we've got two pathways of pyrimidine synthesis and purine synthesis, and then we have degradation pathways for both. The basic, like, high-level kind of summary of the nucleotide metabolism is that we need them so we can synthesize them from simple precursors. When we break them down, we can't get much energy from them. Okay, so nucleotides don't represent a source of stored calories within our body. Okay, as such, we try to not break them down and we try to recycle and reuse them as much as possible. Okay. All right, so what is a nucleotide? Well, nucleotides, functionally, they have diverse functions. We know that they're the subunits that make up nucleic acids, RNA and DNA, but they have a lot of other functions. So cyclic AMP is a second messenger. We know that isocitrate dehydrogenase, for example, that can get activated by ADP. So they're regulatory molecules. We've also seen a few examples in coenzyme A and in nicotinamide adenine dinucleotide, for example, where a nucleotide structure is part of a handle of an organic molecule that allows enzymes to grab it. Okay, so there may be some other functions of nucleotides there, but I think that's a decent summary. Okay, in terms of the structure of a nucleotide, the key structures are a base, and the base is linked through a nitrogen to a 5-carbon sugar, ribose sugar, and then that ribose sugar is linked to a phosphate. Okay, so the three components of a nucleotide are the base, the ribose sugar, and the phosphate. Just in terms of nomenclature, I get this wrong all the time, and you hear these interchanged, but the base is not the same thing as a nucleoside and not the same thing as a nucleotide. So the base, like the adenine base, when that's joined to the ribose sugar, that becomes the adenosine nucleoside. And then if that adenosine nucleoside gets phosphorylated, then it becomes the adenosine monophosphate nucleotide. Is that clear? And if it gets phosphorylated again, it's the adenosine diphosphate. And if it gets phosphorylated thrice, adenosine triphosphate. Okay, but you hear a lot of people talk about the adenine nucleotide or the thymine nucleotide, so cut them some slack, I guess. Okay, so the purine nucleotides are adenine and guanine. These are the ones that are present in DNA. Hypoxanthine, this is a degradation product of the purine nucleotides. Then the pyrimidine nucleotides are cytosine and thymine, the pyrimidine bases are cytosine and thymine for DNA, and then uracil for RNA. Okay, so the basics of the biosynthesis of the nucleotides are kind of conceptually the purines, so the adenine and the guanines, these are built on the ribose sugar part of the nucleotide. They're built atom by atom. Okay, so it starts with the aspartate and then you build it atom by atom, building the base right on the ribose sugar. The pyrimidines, on the other hand, the pyrimidines are building their base separately, or they're building the orotate base separately. Then that whole base gets transferred onto the ribose sugar and then modified further to make either cytosine, thymidine, or uracil. Okay, so the common thing between the purine and the pyrimidine synthesis pathway is this phosphoribosyl pyrophosphate 5-carbon sugar. So let's talk about that PRPP generation. And the PRPP synthetase, the enzyme that is generating that precursor to the sugar portion of the nucleotide, that's a key regulatory step that controls all of the nucleotide biosynthesis pathway. Okay, so the ribose 5-phosphate, this is one of our products of the pentose phosphate pathway. And that ribose 5-phosphate, so ribose 5-phosphate has a single phosphate on the number 5 carbon. The PRPP synthetase enzyme, this is taking ATP and taking 2 phosphates from ATP and adding them onto the number 1 carbon. And then AMP is the leaving group. So this enzyme, this is inhibited allosterically by adenosine diphosphate and guanosine diphosphate, the purine nucleotides. However, the PRPP is used by both purine and pyrimidines. Okay, but the level of control of the PRPP synthetase is by the purine nucleotides. Okay, shall we move on? We'll come back to PRPP, but just wanted to introduce that idea, where it comes from, its linkage to the pentose phosphate pathway, and that it's a key regulatory step. Okay, so let's consider our purines first. So the purine bases are adenine and guanine, and they differ by the position of the amine group and the presence of that keto group. Okay, so as I mentioned before, purine synthesis starts with a molecule of phosphoribosyl pyrophosphate. That then is the base, the purine base is then built atom by atom on the number 1 carbon of the PRPP. So the first addition is a nitrogen that comes from glutamine. So glutamine is deaminated, and the nitrogen is replacing the pyrophosphate on the PRPP. After the nitrogen is bound, then additional carbon from glycine is added, and we get kind of the beginning of the ring structure. So that gets down to, so glutamine plus PRPP, then glycine. Now, there are figures in your textbook, in your Mark's textbook, showing that, this nitrogen comes from glutamine and this part comes from aspartate and all that. And I don't think that's a great use of your time to memorize all that. So just kind of know in general where these things are coming from. So they're coming from, PRPP comes from pentose phosphate pathway. Some of the stuff comes from amino acids, and look at this guy. We just talked about that, right? So that's our most oxidized form of the tetrahydrofolate, the formal tetrahydrofolate that we can get by formate from tryptophan catabolism, right? So that's another carbon donor. We also get carbon from carbon dioxide. Okay, so we're getting all these atoms built one atom at a time from these diverse sources to build this ring. And the common intermediate for both adenosine and guanosine biosynthesis is inosine monophosphate, so shown here. Okay, so once inosine monophosphate is generated, that then can take nitrogen in two different ways from two different sources to make either adenosine monophosphate or guanosine monophosphate. So let's consider the generation of adenosine monophosphate from inosine monophosphate first. And I have a question that's going to lead into that. Okay, let's see. So the argininosuccinate lyase is cutting off fumarate. Very good. And if we recall from our urea cycle, so this is where that reaction happens. So this is a nitrogen transfer reaction that happens in two steps. Okay? So aspartate down here, this nitrogen joins onto citrulline to make argininosuccinate. Then the argininosuccinate lyase is cutting off the non-nitrogen part of aspartate as fumarate, and that leaves then arginine. Okay, so it's a nitrogen transfer or an amination reaction, if you will, in two steps. Now we have a very similar reaction in the conversion of inosine monophosphate to adenosine monophosphate. In this case, we've got aspartate bonding to the keto carbon here through that nitrogen. So this part in pink here is aspartate. Then everything but that nitrogen is going to be cut off as fumarate to leave the adenosine base. Okay, so this biosynthesis pathway of adenosine monophosphate, this is something that we've seen before with our purine nucleotide cycle as an anaplerotic reaction in the TCA cycle. So if you remember the adenosine monophosphate deaminase reaction and myoadenylate deaminase deficiency, remember that condition? Okay, so this is that same reaction. In this case, it's used for de novo synthesis of the nucleotide rather than this cycling of recycling. Okay, so this is that same question. I'm just going to leave this so that the answer is fumarate here. Okay, so to make adenosine monophosphate from inosine monophosphate, we need that two-step amine transfer where we make a covalent bond with aspartate and then fumarate is cut off. To make the guanosine monophosphate, that's still an amine transfer reaction. However, it's a little bit different. It doesn't involve aspartate. So in this case, in the generation of the guanosine monophosphate from the inosine monophosphate, first we're going to oxidize this carbon into a ketone carbon, and then we're going to transfer a nitrogen on it using glutamine as a nitrogen donor. And this is using ATP hydrolysis as an energy source. So for the guanosine monophosphate, we're leaving this as the ketone, and then we've got the amine group down here. Okay, so it seems like a lot of trivia, I guess, but I don't know, should know it, I guess. Okay, so now we have guanosine monophosphate generated and adenosine monophosphate generated. So what happens next then is going to depend on what the molecule is used for. Now both AMP and GMP are going to be phosphorylated to the diphosphate form to make ADP or GDP. What happens after that is going to depend on the fate of that molecule. Now, if that adenosine or guanosine nucleotide is going into RNA, then it will be phosphorylated again to make ATP or GTP. But if it's going to be DNA, then it's got to be modified.
I really wanted to pick bees, but
You did.
I'm doing it.
Okay, so D stands for don't matter. Close, but that's the second best answer. So deoxy, right? So the nucleotides are not equivalent, right? So there are nucleotides and then deoxynucleotides. So in order to convert a nucleotide into a deoxynucleotide, we need an enzyme called ribonucleotide reductase that is converting this alcohol into just a reduced carbon. Ribonucleotide reductase, this is always acting on the diphosphate form. Okay, so if we wanted to make, if we had adenosine monophosphates and we wanted to turn it into deoxyadenosine triphosphates, we would go AMP to ADP, then ADP to deoxy-ADP, then deoxy-ADP to deoxy-ATP. Okay, so ribonucleotide reductase always acts on the diphosphate form, and it's a super easy test question for me to write, so don't be surprised about that. Okay, so where does this reducing power come from to reduce this carbon from an alcohol into a methyl carbon? Comes ultimately from NADPH. We haven't talked about thioredoxins. These are redox proteins, and you remember the cysteine, cysteine, and reduced glutathione and oxidized glutathione. So it's the same kind of idea. So these are proteins that have cysteines that can either be in the oxidized form, making a bond to one another, or in the reduced form. Okay, so they act as electron carriers just like FAD or other redox cofactor. Okay, so the basic idea, the nucleotides are components of RNA, and likewise components of NADH and coenzyme A and some of these other uses for nucleotides. The deoxynucleotides, these are used for DNA. And ribonucleotide reductase is always going to use the diphosphate form as its substrate. Okay. Any questions on purine synthesis? So let's move on then and talk about purine salvage. Now, because we can't get useful energy from the catabolism of nucleotides, the body, and the synthesis of nucleotides is energetically expensive, right? It requires ATP hydrolysis at numerous steps. The body tries to recover and recycle the constituent parts of nucleotides as much as possible. And the purine salvage pathway, this is one of the major pathways of recycling different bits of nucleotides. And this is also a pathway that has a number of inborn errors of metabolism in it. So this again is one of these like master diagrams that I think is useful to have open as you're reviewing this. And I'll kind of walk through how to read it, and then we'll look at some specific examples of it. Okay, so we've got bases over here, nucleosides over here, and nucleotides in the middle. And the goal of this pathway is essentially to get something useful, and the useful things would be the adenosine monophosphate and the guanosine monophosphate. Like those are things that you can actually use to synthesize RNA or to synthesize DNA. So we're trying to get the GMP and the AMP from free bases or from nucleosides, okay? All right, so in order to convert free bases into a nucleotide, we have to add the ribose sugar component to the base. Now in the normal purine synthesis pathway, we build the base atom by atom onto the ribose sugar, right? So this is not a normal way that we would synthesize a base. But if we had a free base around, and if we have PRP around, there is a phosphoribosyl transferase, adenine phosphoribosyl transferase, that can add the base through the nitrogen right onto the ribose sugar to make AMP directly. Okay. Likewise, there is an enzyme that can take the hypoxanthine base and the guanine base and add the phosphoribosyl to that to make the inosine monophosphate or guanosine monophosphate respectively. So this is the hypoxanthine guanosine phosphoribosyl transferase, and this is, again, making the same product, the inosine monophosphate and guanosine monophosphate, but doing it through a base transfer rather than through a building the base up atom by atom. So where would we get these free bases? This could come from like a base excision repair is going to release free bases. So that would be an example of where we might come by these free bases. Okay, so let's see. So the phosphoribosyl transferase enzymes, those are involved in making a free base into a nucleotide. The making a nucleoside into a nucleotide is a little bit dicier or a little bit more complicated. With the adenosine nucleoside, there is a kinase that can directly phosphorylate the ribose sugar component of the adenosine nucleotide to make adenosine monophosphate. So the adenosine base, or excuse me, the adenosine nucleotide, this can be phosphorylated directly. That's not the case with the inosine or the guanosine nucleosides. In this case, the inosine or guanosine nucleosides, they are substrates for a phosphorylase that will remove the ribose sugar completely. And that phosphorylase, as the name implies, that's using inorganic phosphate to have the ribose leave as ribose 1-phosphate. This then, when you take the ribose sugar off of the nucleoside, you're left with the base, and the base can be a substrate for the phosphoribosyl transferase enzyme to make the nucleotide again. Okay, so we don't have an inosine kinase or a guanosine kinase, so that requires them to go through this kind of circuitous pathway of first removing the ribose sugar and then adding the ribose sugar from the PRPP. Okay, does that, I think I might have gotten a little ahead of myself. Okay, is that salvage pathway understandable? Okay, so this is a pathway in which there are numerous inborn errors of metabolism, including a couple of immunodeficiency disorders that, superficially they may look like a immunodeficiency from a folate deficiency, right? Like our case with the patient that had the PCFT mutation. But they have a, different etiology, and we'll discuss that shortly, soon. Okay, so we're going to go through examples of each one of these conditions, so I don't want to get ahead of myself, but there's a list here for you. Okay, so the nucleotide metabolism, we generally try to not do nucleotide catabolism and break them down and urinate them out because we can recycle them and that can save us the energy of synthesizing them. However, we can break down nucleotides when we need to, and we can urinate out excess ones or urinate out the products. Now, we don't get energy from this. However, if we are breaking down cells and breaking down DNA, you need to get rid of them under some circumstances. So this is the way that we break down the purines. So the purine, let's see, the GMP and the AMP, these are both broken down to the intermediate xanthine, and then xanthine can be converted into uric acid, which is urinated out. So the guanosine monophosphate, so not shown here is the ribose sugar, but the first step in its catabolism is to remove the phosphates from the 5-carbon of the ribose sugar to make the nucleoside guanosine. Then a phosphorylase removes the ribose sugar using inorganic phosphate to remove the ribose as ribose 1-phosphate. Okay, then we're left with the guanine base. The guanine base is then deaminated to xanthine, and then xanthine, gesundheit, is then oxidized by xanthine oxidase at the carbon on the beak here to make uric acid. And uric acid is ionizable, near enough to physiological pH that it can be soluble and urinated out. So with the AMP, the metabolism is a little bit different, or catabolism is a little bit different. We're losing the nitrogen first, then dephosphorylating the base, and then getting rid of the ribose sugar to make the hypoxanthine. The hypoxanthine is oxidized at this carbon to make xanthine, and then we go through the same oxidation at this carbon to make uric acid. Interestingly, the oxidations of hypoxanthine and xanthine, these are both carried out by the same enzyme. So xanthine oxidase is carrying out both of these reactions. And let's see, had we talked about reperfusion injury at all? No, I don't think so. A little bit? Okay.
That was a different class.
I think I might be getting you mixed up with the second years. So, all right, sorry about that. Okay, so there are some places that this can go wrong as well. Uric acid, if we get excessive nucleotide catabolism, we can get a lot of uric acid built up faster than we can excrete it. This can crystallize, and this can cause gout if the crystals are in the extremities, or it could cause a urinary or a kidney stone if it happens in the kidney.
What causes the excess degradation of the nucleotides that would cause that?
The excess degradation of nucleotides can be caused by eating a lot of nucleotides, is one thing. So if you eat a lot of organ meat, you're eating a lot of nucleotides and you're catabolizing a lot of them. So that's definitely something that can increase the nucleotide catabolism and contribute to gout, just your diet. You can also get it anytime you have like tissue injury or tissue death. That tissue has got DNA in it, and that DNA can get broken down into the constituent parts and broken down into uric acid. Now, in that situation, that's going to probably be like an acute, one-time thing, so it's not going to result in gout necessarily, but it could result in hyperuricemia. There's a condition that we'll see in a little bit called Lesch-Nyhan disease, and that's a disorder in the purine salvage pathway that results in hyperuricemia and gout, and yeah. So there's a few like ways you can get it through kind of obvious sources by eating a lot of nucleotides or salvage pathway disorder. You can also get it just by growing old and kind of idiopathic gout. So, okay. Any other questions? Looks like we're right near the break, so maybe we should take a break. We'll take a break and then we'll talk about pyrimidines. Pyrimidines are quite a bit simpler than the purines, thankfully.
Or it's Dunzo. That was the last lecture. I
I asked you to do it, and you really
Is this what you wanted from me? Am I making you proud?
that
I knew it was gonna happen though. Because since they've got used to not being on the day. No, I think it's just last year's.
Because I'm walking by on the left side, I was just like.
So then you just like pick up.